Some discoveries announce themselves with thunder. Others arrive in silence. On a quiet morning in 1991, Ann Tsukamoto stood in her laboratory at SyStemix in Palo Alto, California, watching fluorescent markers dance across her computer screen. Each glowing dot represented a single cell, sorted from millions of others in human bone marrow. But these weren't ordinary cells. These were the master cells, the ones that could become anything the blood needed. For decades, doctors had wielded chemotherapy like a sledgehammer against cancer, destroying malignant cells along with the bone marrow that sustained life itself. They could kill the disease, but they couldn't rebuild what they'd shattered. Tsukamoto had just solved that problem. The cells glowing on her screen were human blood stem cells, isolated and identified for the first time in history. What flowed from this moment would give doctors the power not just to destroy cancer, but to heal what remained. Ann Tsukamoto was born in 1952 in California, a child of the post-war era when America was still reckoning with what it had done to its own citizens. Her parents were Japanese Americans who had lived through the internment camps of World War II, held behind barbed wire simply for their ancestry. They rarely spoke of those years, but the experience shaped everything about how they raised their daughter. Education became sacred. Achievement became armor. Excellence became the only acceptable standard. Growing up in the 1960 and 1970, Anne inhabited multiple worlds that rarely intersected. At home, she lived within Japanese cultural traditions that emphasized duty, precision, and quiet determination. At school, she navigated an America where girls were steered toward teaching or nursing, and Asian faces remained rare in science classrooms. The combination should have discouraged her from pursuing research science. Instead, it forged something stronger. Her path to biology wasn't inevitable. As a young student, Anne excelled in multiple subjects, but something about cellular biology captured her imagination in a way nothing else had. Perhaps it was the hidden complexity, the way microscopic structures contained entire worlds of function and possibility. Perhaps it was the precision required, the careful observation that matched her training, or perhaps it was simply that cells didn't care about race or gender. They revealed their secrets to anyone patient enough to look closely. At the University of California, San Diego, Anne pursued her undergraduate degree in biology at a time when women made up less than 10% of science doctoral programs. She encountered professors who questioned whether women had the temperament for rigorous research. She faced assumptions that Asian students were good at memorization, but lacked creativity for original investigation. These obstacles didn't deter her. If anything, they sharpened her focus. She would let her work speak for itself. Anne's graduate studies at UCLA brought her deeper into immunology and cellular biology. She became fascinated by the immune system's ability to generate diverse cell types from common precursors. Blood cells particularly intrigued her with their variety and specialization. Somewhere in bone marrow, she learned, lived the master cells that could become red blood cells, white blood cells, or platelets. Finding and isolating these stem cells became the holy grail of hematology, a problem that would define her career. Every 3 seconds, someone in America receives a diagnosis that will change everything. Leukemia, lymphoma, multiple myeloma, blood cancers that transform the body's life-sustaining fluid into a weapon against itself. By the time Ann Tsukamoto entered the field in the early 1980s, medicine had developed powerful chemotherapy drugs that could destroy these malignant cells. But the treatment came with a terrible price. Chemotherapy didn't distinguish between cancerous blood cells and the healthy stem cells in bone marrow that produced them. To kill the cancer, doctors had to devastate the entire blood-forming system. Patients who survived the initial treatment faced months of vulnerability, their bodies unable to fight infections or stop bleeding. Many died not from the cancer itself, but from complications during the long, uncertain recovery period when their bone marrow struggled to regenerate. The solution seemed obvious in theory. If doctors could isolate healthy blood stem cells before treatment, preserve them, and return them to patients after chemotherapy, they could help rebuild destroyed bone marrow. The practice, however, proved maddeningly elusive. Stem cells represented perhaps 1 in 10,000 cells in bone marrow, invisible needles in a haystack of ordinary blood cells. No one knew exactly what they looked like or how to separate them from everything else. Scientists had identified stem cells in mice, but human biology presented different challenges. Mouse experiments could be controlled and replicated in ways impossible with humans. Researchers could breed mice with specific genetic markers and track cells through multiple generations. Human stem cells remained theoretical, their existence inferred from observation, but never directly confirmed. Without being able to isolate and study them, scientists couldn't develop reliable transplant protocols. The stakes extended beyond cancer treatment. Understanding blood stem cells could revolutionize treatment for genetic blood disorders, autoimmune diseases, and immune deficiencies. Sickle cell anemia, thalassemia, and severe combined immunodeficiency might become curable if doctors could replace defective stem cells with healthy ones. The potential applications seemed limitless, but everything depended on solving the fundamental problem of identification and isolation. Research labs worldwide pursued the challenge throughout the 1980s. Scientists tried various approaches using different cell markers and separation techniques. Progress came in frustrating increments. A technique would show promise in preliminary experiments, then fail to work consistently. Markers that identified stem cells in mice didn't translate to humans. The cells were there, hidden in plain sight, but they refused to reveal themselves. Ann Tsukamoto joined this global hunt, bringing her training in immunology and her characteristic patience to a problem that had defeated countless researchers before her. Ann Tsukamoto arrived at SyStemix in the mid-1980s, joining a small biotechnology company founded on an audacious premise that human blood stem cells could be found, isolated, and turned into medicine. The company occupied modest laboratory space in Palo Alto, part of the biotech boom transforming Silicon Valley from semiconductor center to life sciences hub. While other companies chased blockbuster drugs, SyStemix pursued something more fundamental, the basic biology that could reshape entire fields of treatment. The technical challenge was staggering. Bone marrow contained hundreds of different cell types, all mixed together in a complex cellular soup. Blood stem cells looked similar to many other cells under a microscope, offering no obvious visual markers. They divided slowly, making them difficult to track. They existed in tiny numbers, requiring researchers to sort through millions of cells to find a handful of candidates. And there was no reliable test to confirm whether a cell was truly a stem cell or just another ordinary cell that happened to share some characteristics. Tsukamoto and her colleagues developed a methodical approach based on cell surface proteins. Every cell displays specific proteins on its outer membrane, like identification badges announcing its type and function. Using fluorescent antibodies that bound to particular proteins, researchers could tag different cell types with different colors. A flow cytometry machine could then analyze thousands of cells per second, sorting them based on their fluorescent signatures. The question was which proteins marked stem cells. The research proceeded through endless cycles of hypothesis and testing. The team would identify a promising protein marker, tag cells expressing it, isolate them, and test whether they behaved like stem cells in laboratory cultures. Most attempts failed. Cells that looked promising would fail to generate diverse blood cell types. Markers that worked partially would capture too many non-stem cells along with the targets. Each failure provided information, narrowing the possibilities, but the goal remained elusive. Pressure mounted from multiple directions. Competing research groups at major universities pursued similar approaches, racing to publish first. Cancer patients continued dying from transplant complications that better stem cell isolation might prevent. Investors wanted results that could translate into therapies and revenue. Scientific journals rejected papers that showed interesting but incomplete findings. The slow, grinding nature of the work tested everyone's resolve. Tsukamoto brought a particular temperament to these challenges, where others might rush to publish preliminary results or chase more fashionable research directions. She remained focused on getting the fundamental biology right. Her training had taught her that precision mattered more than speed, that a reliable method based on solid science would ultimately prove more valuable than dramatic claims built on shaky evidence. She refined techniques, repeated experiments, and insisted on rigorous controls that some colleagues found excessive. The breakthrough, when it finally came, emerged from combining multiple markers rather than seeking a single identifying protein. Tsukamoto and her team realized that stem cells might be defined not by what they expressed, but by a specific combination of presence and absence. By looking for cells that displayed certain proteins while lacking others, they could narrow the pool, dramatically isolating a population where stem cells concentrated. The data appeared on Tsukamoto's computer screen on an ordinary Tuesday morning in 1991, but there was nothing ordinary about what it revealed. After sorting bone marrow cells using their carefully developed combination of markers, the team had isolated a tiny population representing less than 1% of the original sample. They cultured these cells in laboratory dishes designed to support blood cell growth. Then they waited. Over the following weeks, those isolated cells did something remarkable. They divided and differentiated, generating red blood cells, multiple types of white blood cells, and platelet, producing megakaryocytes. The cells didn't just become one type of blood cell, they became all the types, exactly as true stem cells should. Tsukamoto ran the experiment again, refining the protocol. The results held they had found human blood stem cells. The technical achievement rested on identifying a specific combination of cell surface markers. The critical cells were CD34 positive, meaning they expressed the CD34 protein on their surface, but they were also negative for other markers associated with mature blood cells. This combination of presence and absence created a signature that could be detected by flow cytometry and used to sort stem cells from the complex mixture of bone marrow cells. What made the discovery transformative was its reproducibility. Previous researchers had occasionally isolated cells that seemed to have stem cell properties, but they couldn't reliably repeat the process or scale it to practical applications. Tsukamoto's method worked consistently. Different technicians could follow the protocol and achieve similar results. The technique could process clinically relevant amounts of bone marrow. It was science that could leave the laboratory and enter hospitals. The patent application filed in 1991 represented more than protecting intellectual property. It codified the exact method for isolating human blood stem cells, creating a blueprint that other researchers could follow and build upon. Patent number 50,61,600. Would become one of the most cited patents in stem cell biology, referenced in thousands of subsequent research papers and clinical applications. Within the laboratory, the team celebrated with cautious excitement. They understood the magnitude of what they had achieved, but they also recognized the work still ahead. Isolating stem cells in a laboratory was one thing. Developing safe, effective clinical protocols for transplanting them into patients was another. They had solved a fundamental problem, but translating that solution into medical practice would require years of additional research, clinical trials, and regulatory approvals. News of the breakthrough spread through the scientific community with remarkable speed. Other research groups immediately began testing Tsukamoto's method, confirming her results and exploring variations. Pharmaceutical companies recognized the commercial potential of stem cell-based therapies. Cancer treatment centers saw possibilities for improving transplant outcomes. The discovery had opened a door, and researchers worldwide rushed through it, each pursuing different applications of the fundamental technique. For Tsukamoto personally, the moment brought vindication but little rest. She had proven that a Japanese-American woman could make foundational contributions to medical science. She had demonstrated that patient, methodical research could solve problems that had defeated flashier approaches. But she also knew that this was just the beginning. The real measure of her work would be whether it actually saved lives. The first patient to benefit from Tsukamoto's work never knew her name. In a hospital somewhere in America, a leukemia patient received a stem cell transplant using cells isolated according to the protocols she had developed. The transplant engrafted successfully. The patient's bone marrow recovered faster than expected. Doctors reported the results in a medical journal, citing Tsukamoto's patent among dozens of references. The patient went home to their family, carrying stem cells that had been found using techniques perfected in a Palo Alto laboratory. This pattern repeated thousands of times as the 1990s progressed. Cancer treatment centers worldwide adopted CD34 positive cell selection as standard practice for stem cell transplantation. The technique allowed doctors to harvest stem cells from patients before chemotherapy, isolate the valuable cells from contaminating cancer cells or mature blood cells, and return a purified population that could rebuild the bone marrow more efficiently. The medical impact extended beyond blood cancers. Patients with genetic blood disorders became candidates for stem cell transplants that might cure conditions they had been born with. Children with sickle cell anemia could potentially receive healthy stem cells that would generate normal red blood cells for the rest of their lives. Adults with severe autoimmune diseases found hope in transplants that could reset their immune systems. The applications multiplied as doctors gained confidence in the technique. One early success story involved a young woman with aggressive lymphoma who had failed multiple rounds of conventional chemotherapy. Her oncologist proposed an autologous stem cell transplant, harvesting her own stem cells, treating her with high-dose chemotherapy that would destroy the cancer along with her bone marrow, then rescuing her with the preserved stem cells. The stem cell harvest used Tsukamoto's CD34 selection method, isolating millions of pure stem cells free from contaminating lymphoma cells. The woman survived the brutal chemotherapy regimen, her body sustained by transfusions while her bone marrow remained devastated. 3 weeks after receiving her own purified stem cells back through an intravenous line, her white blood cell counts began rising. By week 4, she was producing platelets. Her bone marrow had regenerated faster and more completely than with previous transplant techniques. 5 years later, she remained cancer-free. Her body built on blood cells descended from those isolated stem cells. Stories like this transformed stem cell transplantation from a desperate last resort to a planned therapeutic strategy. Survival rates improved as techniques refined. Doctors could now harvest more stem cells with less contamination. They could store them safely for months. They could monitor engraftment by tracking CD34 positive cells in patients' blood. The entire field of transplant medicine evolved around the fundamental ability to reliably identify and isolate the cells that mattered. The economic implications were equally significant. Before reliable stem cell isolation, bone marrow transplants required extensive hospitalization, carried high complication rates, and cost hundreds of thousands of dollars. Improved techniques reduced hospital stays, decreased infections and bleeding complications, and lowered overall treatment costs. Insurance companies began covering stem cell transplants for conditions they had previously considered too experimental. The medical procedure became more accessible, even as it became more effective. Tsukamoto continued her research throughout this period, refining isolation techniques and exploring stem cell biology. She published papers describing improved protocols, addressing technical challenges that arose in clinical practice, and investigating what made some stem cells more effective than others at regenerating bone marrow. Her work remained focused on the fundamental questions, letting others pursue the commercial applications, while she ensured the science stayed rigorous. The discovery of how to isolate blood stem cells created opportunities that extended far beyond treating cancer. If stem cells could be found, frozen, and transplanted, then they could be collected, stored, and distributed. What emerged was an entirely new medical infrastructure built around matching stem cells from healthy donors to patients who needed them, a system that continues saving lives decades after Tsukamoto's breakthrough. Bone marrow registries had existed before, maintaining databases of volunteers willing to donate marrow to strangers with matching tissue types. But the process was painful, requiring surgical extraction of marrow from hip bones under anesthesia. donation rates remained low despite the need. Tsukamoto's work enabled a revolution in how stem cells could be obtained. Researchers discovered that mobilizing stem cells from bone marrow into circulating blood, then collecting them through apheresis, provided a less invasive alternative. The protocol involved giving donors medication that stimulated stem cell production and release into the bloodstream. After a few days, the donor's blood was processed through a machine that separated out the CD34 positive stem cells while returning everything else. The donor experienced something closer to giving blood than undergoing surgery. This advancement dramatically expanded the potential donor pool, making stem cell donation accessible to more people and reducing the barriers to matching patients with unrelated donors. Umbilical cord blood emerged as another source of stem cells, one that had been routinely discarded as medical waste until researchers recognized its value. After birth, the blood remaining in umbilical cords and placentas contains rich concentrations of stem cells. Hospitals began collecting cord blood, processing it using Tsukamoto's techniques to isolate the stem cells and freezing the samples in specialized banks. Each unit represented a potential life-saving transplant for a child or small adult. Public and private cord blood banks proliferated throughout the 1990s and 2000s, building inventories that could serve patients worldwide. The National Marrow Donor Program expanded to include cord blood units alongside volunteer donors. International registries connected, allowing doctors to search millions of potential matches for their patients. The infrastructure grew sophisticated, with standardized testing, quality controls, and shipping protocols that could deliver frozen stem cells to hospitals anywhere in the world within days. Tsukamoto played an advisory role in this expansion, consulting with organizations establishing stem cell banks and registries. Her expertise in stem cell biology helped shape quality standards and collection protocols. She advocated for rigorous testing to ensure that collected stem cells met minimum standards for count and viability. Her voice carried weight in debates about banking practices, transplant techniques, and research priorities. The growth of stem cell banking also created new ethical questions. Private cord blood banks marketed to expectant parents, charging thousands of dollars to store their baby's cord blood for potential future use. Public banks collected donations for general use. Debates erupted over whether private banking represented prudent preparation or unnecessary expense, exploiting parental anxiety. Tsukamoto weighed in, emphasizing that most families would never need their stored cord blood, but that public banking served clear medical and social good. Training programs emerged to educate the next generation of stem cell researchers and clinicians. Tsukamoto participated in seminars and workshops, teaching techniques and sharing hard-won knowledge about what worked in practice versus what looked good on paper. She mentored graduate students and postdoctoral researchers, many of them women and minorities who saw in her an example of what they might achieve. Her laboratory at SyStemix, and later at other institutions, became a training ground for leaders in stem cell medicine. By the early 2000s, the infrastructure Tsukamoto had helped build supported thousands of stem cell transplants annually. Registries contained millions of potential donors. Cord blood banks held hundreds of thousands of units. Hospitals had established specialized transplant centers with teams trained in the latest protocols. What had begun as a laboratory technique had become a global medical system, a network of knowledge, materials, and expertise, all tracing back to the fundamental ability to identify and isolate human blood stem cells. Tsukamoto's breakthrough with blood stem cells opened floodgates in regenerative medicine. If blood-forming stem cells could be isolated and characterized, perhaps other tissues harbored similar master cells. Researchers began hunting for stem cells in brain tissue, muscle, skin, and organs. The 1990s and early 2000s became a gold rush era for stem cell biology, with new discoveries announced regularly and implications that seemed to expand exponentially. The excitement reached fever pitch in 1998, when researchers isolated human embryonic stem cells, pluripotent cells capable of becoming any tissue in the body. The discovery promised revolutionary treatments for conditions from spinal cord injuries to Parkinson's disease, but it also ignited fierce ethical debates. Embryonic stem cells came from embryos, raising profound questions about the moral status of early human life and the ethics of destroying embryos for research purposes. Ann Tsukamoto watched these developments with a scientist's fascination and a pragmatist's caution. She understood the potential but had seen too many promising leads fail in practice to embrace hype uncritically. She advocated for continued focus on adult stem cells, the tissue-specific cells like those she had worked with, which raised fewer ethical concerns while offering proven medical applications. She worried that overblown claims about embryonic stem cells would create unrealistic expectations and potential backlash that could harm the entire field. Her instincts proved prescient media coverage of stem cell research often blurred the line between theoretical possibility and clinical reality. Patients with devastating conditions heard promises of miracle cures that remained decades from practical application. Some sought treatments at offshore clinics offering unproven stem cell therapies, sometimes suffering harmful consequences from procedures that lacked scientific foundation. The field needed voices of reason, scientists who could communicate both the promise and the limitations. Ann Tsukamoto became an advocate for responsible science and realistic communication. She gave presentations to medical groups, patient advocacy organizations, and policymakers, explaining what stem cell medicine could actually deliver versus what remained speculative. She emphasized that reliable treatments required years of rigorous testing, that promising laboratory results often failed to translate to clinical success, and that patients deserved honesty about timelines and uncertainties. The political landscape around stem cell research grew contentious during the early 2000s. Federal funding restrictions on embryonic stem cell research created fierce debates about scientific freedom, religious values, and government's role in regulating research. California voters passed Proposition 71 in 2004, creating a $3 billion state-funded stem cell research initiative. Tsukamoto participated in discussions about how those funds should be allocated, advocating for balanced support of both basic research and clinical translation. Meanwhile, work continued on applications of adult blood stem cells, the area where Tsukamoto's contributions remained most relevant. Researchers explored using stem cells to treat autoimmune diseases like multiple sclerosis and lupus. Clinical trials investigated whether resetting the immune system through stem cell transplantation could halt disease progression. The results showed promise for carefully selected patients, though the treatments remained intensive and carried significant risks. Tsukamoto's own research evolved to address more subtle questions about stem cell biology. Not all stem cells performed equally well in transplants. Some patients' stem cells engrafted quickly while others struggled. Understanding what made certain stem cells more potent or long-lived could improve treatment outcomes. She investigated the molecular characteristics that distinguished robust stem cells from less effective ones, publishing findings that helped optimize collection and transplant protocols. The stem cell field also grappled with commercial pressures and intellectual property disputes. Tsukamoto's original patent became extraordinarily valuable as stem cell therapies expanded. Companies developing stem cell products licensed the technology, generating revenue but also creating tensions around access and affordability. She navigated these dynamics carefully, believing that scientific discoveries should benefit patients broadly, while recognizing that commercial development required investment and protection of intellectual property. The National Inventors Hall of Fame inducted Ann Tsukamoto in 2018, placing her alongside Thomas Edison, the Wright brothers, and other transformative inventors. The ceremony celebrated her work isolating human blood stem cells, acknowledging that her discovery had saved countless lives and generated billions in economic value. For someone who had spent decades in laboratories, the public recognition represented a remarkable moment. Yet, even with this honor, most people still didn't know her name or understand her contribution. Tsukamoto's patents, particularly the foundational 1991 patent on stem cell isolation, became among the most cited in biotechnology. She holds 12 patents related to stem cell biology, creating an intellectual property portfolio that shaped how stem cell medicine developed commercially. These patents generated substantial licensing revenue for her employers and established legal frameworks that other researchers navigated as they developed new applications. Professional recognition came steadily throughout her career. She was elected to the American Association for the Advancement of Science, joining an elite group of researchers whose work had significantly advanced scientific knowledge. She received awards from medical societies, patient advocacy organizations, and biotechnology associations. Each recognition acknowledged not just the initial breakthrough, but her sustained contributions to translating laboratory discoveries into clinical practice. The statistical impact of Tsukamoto's work appears in cancer survival data. 5-year survival rates for acute myeloid leukemia, one of the deadliest blood cancers, improved from approximately 20% in the 1980 to over 30% by the 2020 for adult patients, with even better outcomes for children. Advances in stem cell transplantation contributed significantly to these improvements. Patients who once faced near certain death now had fighting chances, sustained by transfusions of carefully isolated stem cells. Bone marrow transplant registries demonstrate the scale of impact. The National Marrow Donor Program, now called Be the Match, has facilitated over 100,000 transplants since its founding. Worldwide, hundreds of thousands of patients have received stem cell transplants using techniques that rely on Tsukamoto's methods for identifying and isolating the critical cells. Each transplant represents a life potentially saved, a family spared grief, a future made possible by understanding how to find the cells that matter. Current medical practice incorporates stem cell isolation so thoroughly that practitioners rarely think about its origins. When an oncologist orders a stem cell harvest, when a laboratory technician runs a CD34 count, when a transplant coordinator verifies that collected cells meet minimum standards, they're applying knowledge that flowed from Tsukamoto's work. The techniques have become standard of care, unremarkable in their routine application, yet revolutionary in their impact. Tsukamoto continued working in biotechnology research long after her initial breakthrough, contributing to companies developing next-generation stem cell therapies and diagnostic tools. She remained active in scientific conferences, presenting data on stem cell biology and engaging with researchers pursuing new frontiers. Her presence in these forums provided continuity, connecting pioneers who had built the field to younger scientists carrying it forward. The demographic significance of Tsukamoto's success extends beyond her individual achievements. As a Japanese-American woman in science, she broke barriers and created pathways for others. Asian American representation in STEM fields has increased substantially since the 1970s, though challenges persist. Women have made gains in biological sciences, though leadership positions remain disproportionately male. Tsukamoto's career demonstrates what becomes possible when talent meets opportunity, regardless of gender or ethnicity. Yet relative anonymity persists outside specialized circles. Ask most people to name a stem cell researcher, and they might mention Shinya Yamanaka, who won a Nobel Prize for induced pluripotent stem cells, or struggle to name anyone. Tsukamoto's work, while foundational, lacks the drama of embryonic stem cell controversies, or the revolutionary narrative of turning adult cells back into stem cells. Her contribution was quieter, more technical, but no less transformative in its practical impact on human health. Ann Tsukamoto's story defies easy categorization. She didn't cure cancer or invent a blockbuster drug. She didn't generate political controversy or capture public imagination. What she did was solve a fundamental problem with extraordinary precision, creating a technique that enabled others to heal. Her legacy lives in every cancer patient who survives, because doctors could safely destroy and rebuild their bone marrow, in every child born free of genetic blood disorders that should have defined their lives. The characteristics that made Tsukamoto successful: methodical patience, rigorous attention to detail, willingness to persist through failures represent precisely the qualities that effective science requires. Breakthroughs rarely arrive in eureka moments. More often, they emerge from accumulated expertise and problems approached from slightly different angles until something finally clicks. Her work continues inspiring new generations of researchers, particularly women and minorities, who see in her career proof that scientific achievement transcends demographic categories. The stem cell medicine field she helped establish keeps expanding, with researchers developing treatments for conditions that remain incurable today. The millions of lives saved by stem cell transplantation represent Tsukamoto's most profound legacy. The woman who taught doctors to grow healing cells reminds us that the most powerful contributions often come from those who focus on the work itself, letting results speak louder than recognition.